This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 470, recorded on December 1st, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. Get $30 off your first delivery and free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
It's gorgeous out.
It's fabulous.
Do you have any sense of the thermal gradients?
It's about 54.5 degrees Fahrenheit.
How about Celsius? I'm
going to try to convert that here. It's got to be somewhere below 21, so that's about maybe 18?
11.
Oh.
11 Celsius.
Way off.
Mostly sunny. Very nice.
Mostly sunny, patchy, little fluffy clouds. Almost no contrails. So that means there's not a lot of moisture in the air. I don't know. Maybe that's what it means.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Hey, Rich.
Well,
we got 73 Fahrenheit, 23 centigrade, 40% humidity, 2.47 degrees, and it's a cloudless day. There's been a whole string of these. It's just gorgeous.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
Here it's 46 degrees Fahrenheit, 8 degrees Celsius, and blue skies, sunny, but with some wispy clouds.
Yeah. And from western Massachusetts, Ellen Dove.
All of the clouds are up here. You can come take some.
Oh, really?
Yeah. It's
too bad.
So it's 50 Fahrenheit, 10 Celsius, and getting some gusty wind, and we've got an overcast, a broken layer at about 3,000, 4,500 feet. So really cloudy and gray looking now.
Wind's out of the...
let's see, wind is out of the north.
I just wanted to mention this morning, driving into work, I drive up the New Jersey Turnpike.
Which exit?
Which is famous from a Simon and Garfunkel song, right?
Yes, the song runs through my head the minute you mentioned
That's
that.
right.
We've all come to search for America, right?
We've
That's
all
right.
come to search for America. By the way, speaking of singing, it's Kathy's birthday today. Happy birthday, Kathy.
Happy
Hey,
birthday.
Birthday,
nicely
Kathy.
done.
Thank you.
Nicely done.
Um,
Keep respiring.
I will.
Thank you.
December 1st. And I saw a bald eagle in a tree next to the New Jersey Turnpike.
Nice.
Usually I see other kinds of hawks,
Right.
but this I could tell, the white.
Yeah.
It was just so cool.
Wednesday, I went trout fishing in Pennsylvania, and I saw one too. It flew right
But here,
down the river.
the Turnpike is weird to see one.
They're scavengers, and there's a lot of cars on the New Jersey Turnpike, so therefore there's a lot of carrion, and therefore they get a lot to eat.
Well, the Turnpike goes through the Meadowlands,
Yes,
which
it
is
does.
a marshy area of New Jersey. There's a lot of development there, but there's also
true.
land that you cannot develop, I presume, because
That's right.
it's too marshy.
Oh, you're right.
The cool thing about it, there are lots of reeds and trees, and there's a lot of wildlife in there.
That's true.
And you can see the tidal shifts. Sometimes when I'm driving past, there's hardly any water, and then sometimes it's very flooded.
So if you, as your next trip down the Jersey Turnpike, especially in the morning, you can see red-tailed hawks.
Yes, of
and
course.
they're positioned, and they space themselves almost to the millimeter. They each have a territory that they feed on,
Yep.
and they're all equally distributed throughout the entire Jersey Turnpike. It's quite amazing.
Well, I can tell you that twice a day I travel the New Jersey Turnpike,
Right.
without fail usually,
It will be harder to see them.
except on weekends.
Right. Right.
Oh, boy. if I only lived 10 minutes from home.
Well, I do.
I know. You're very lucky.
Live 10 minutes from home or work
That
10
would be
minutes?
very inconvenient.
I live at work.
Kathy, that's called a Freudian slip.
I live at work, I guess, yeah.
Exactly right.
I've never, except in grad school, I was 10 minutes from grad school.
Right.
Even my postdoc was not. Although Boston, I was a 20-minute subway ride. That wasn't so bad.
So here's a question for everybody. Do cross-dressers wear Freudian slips?
Where do you get your jokes from?
I don't know.
right, we have some
I
follow-up.
think they come wrapped around a piece of bubble gum usually.
That's true. Bazooka.
We have some follow-up. First is from Islam. Just wanted to say thank you for recording a TWiV episode on the Friday after Thanksgiving when pretty much everybody else was holidaying. Thanks for making my Mondays wonderful. He sent a picture of his dashboard in his car, and he was playing TWiV. I think it's so cool that you can... well, of course, you can play anything on if you have one of these in-dash systems. I don't have one in my car, but you can. Yeah. And then it's got TWiV 469 mycovirus stuffed potatoes. It's cool. By the way, I love Rich's statement about his career in science. Respect.
Indeed.
Please tell Dickson that I printed out the 8 flu segments on a 3D printer already with help from a brilliant PhD student in my previous group at MIT. They are not magnetic, though, but it is fun shuffling them around to mimic reassortment. And he sent a picture of this.
Cool.
Dickson, you need one of these.
I would love one of those. Sure.
Oh, these are pretty cool. I like these.
If you could engineer them with Velcro ends, you could shuffle them up and definitely get reassortment to occur.
Dickson, how many segments per virion would you need to get an infectious particle? It's not the same as how many genome types there are.
6?
It's different. No.
I don't know.
Well, you could just count them in the picture.
1, 8.
Yeah. I tried, Dickson.
Well, you don't succeed.
Alan, can you take
It's okay, Dickson. You can look it up.
Exactly right.
He doesn't look it up.
No.
Alan, can you take the next one, please?
Sure. Ed Ribicki writes, I'm convinced there are a lot of fungal and plant viruses that do the crossover thing. Plants were the obvious hosts for fungi when both crawled out onto dry land. It's obvious that they would have been able to exchange viruses. And I note this, quote, endoRNA viruses, genus Endornavirus, are double-stranded RNA viruses that infect plants, fungi, and oocytes, which have recently been given family status. And I don't know what he's quoting. TMV has been shown, tobacco mosaic virus has been shown to replicate in fungi. BMV, brome mosaic virus, replicates in yeast. Certain animal viruses, e.g. flockhouse virus, can replicate in plant cells but not spread. It's not unusual, is what I'm saying, and we will find a lot more evidence for it.
Yes, this was all mentioned in the podcast.
It does sound like a reasonable projection.
But that paper was very good. It was a very comprehensive analysis of a cross-kingdom infection, and they did say that it's not the first, but it's not very. As far as we know, it's rare. But as Ed said, it's probably not so rare. But
it's rarely documented so far.
Yeah,
rare is definable by that. We haven't looked enough yet.
Yeah, exactly. Kathy, can you take the next one?
Sure. Diane writes, "Hello, Professors TWiV. Thank you for a wonderful show. I look forward to each episode. I enjoy the group dynamics, and so I'm always a little bummed when it is a travel episode. That is, I'm disappointed until I start listening, and then I'm so fascinated by the various interviewees that I end up loving those episodes too. When you first started your Patreon account, I immediately signed on, for I have gotten so much pleasure from listening to all the Twix podcasts. It's definitely worth paying for. In regards to the discussion of long working hours, I would love to hear from Kathy and other female PIs. John Udell and his son both said that in order to be successful in the lab, you need a good wife, or words to that effect. That was so off-putting for me. I would be interested in hearing about how your spouses managed their own careers. Vincent, you have mentioned many times that your wife is a scientist as well. Do you mind talking about the logistics of raising a family with 2 working scientists? How many spouses, what percentage of PIs have intense careers of their own? Does marrying a passionate scientist often mean giving up on one's own career? I think it has been established that in 2-income families, the wife tends to do disproportionately more work around the home. That must include female PIs. How have they managed? In any event, the discussion is fascinating, and I look forward to hearing more. As for me, I'm an oncology chemotherapy nurse. I don't remember how I stumbled onto TWiV, but I did, and I have gotten hours and hours of pleasure listening and learning. Many times the papers are far above my head, but there's enough other stuff that I can't wait to tune in every week. I also listen to TWIP, TWIM, and the thrilling new Immune. Love that show. I also listen to various oncology-related podcasts, The Skeptic's Guide to the Universe, The Gist, Stardate, Still Processing, On the Media, and The Kicker. Sometimes I can't stay current with all these podcasts. Thank you for a great family of podcasts. Diane, who's in Seattle. P.S. I've been meaning to write to TWiP as well because I listened to the entire back catalog. When I was listening to episode 100, my husband walked in right as Dickson started a sentence saying, "As a teacher of parasites." And without missing a beat, my husband said, I wouldn't think parasites would need a teacher. Da-da-da. Thought you would enjoy. Thanks again.
Goodness gracious. All right, I'll try to rephrase that. And she shouldn't feel bad about not understanding the papers. Neither do I.
So back to her question about hearing from me and other female PIs. So I'm not going to be the one to tell you about how it is to have a spouse because I don't have one. I don't have kids. And so I can't really relate to that. But I can tell you that many times when I was writing grants, I would say, oh, man, I wish I had a spouse who could be cooking and cleaning for me right now. And it is the case that I'm not sure how broad the net is, whether it's biology or biomedical sciences or science in general, but more women have spouses who are scientists than men scientists have women scientists who are spouses, if that makes sense. So that is an issue, and it would be good to hear from some other women about how they manage their careers. And then we can hear from you guys about how your spouses managed your careers.
So do you work 80 hours a week without any problem?
No, heavens no.
No, you don't?
No, and I've often said that if I didn't have music to take me out of the lab at night, I would probably stay and work longer, but I don't think I would be that much more productive. So, I have music lesson, I have 2 different rehearsals in the average week. And then performance weeks like this one, I have something every night. And so, I think people with kids, their kids draw them out of the lab and back to home. But in my case, it's music.
Well, I'm trying to get my wife on the show so she can talk about her career.
Cool.
I should probably footnote what I said by saying that while I was in graduate school and postdoc, my wife had a job. She was a technician while I was in graduate school. And wow, when I was a postdoc in England, she, since she has dual citizenship, having been born in London, she worked in the greenhouses in Hyde Park.
Oh,
it was
nice.
Awesome. Yeah, it was great. But once we had kids, she chose to stay with them. And so she was a full-time mom. And of course, that made things a lot easier for me.
I'll let my wife speak. I don't want to talk for her.
Good idea. A very good idea.
Well, she's not so interested in coming on, but I'll try and talk her into it. I keep telling her people are asking for her, so maybe eventually it'll convince her.
Well, and Doris would be really good to have on TWiP, right?
Yeah, she could do TWiP. Yeah, ivermectin and all that.
You bet.
I mean, I want her to come on TWiP as well.
I'm thinking of recording her and putting her on all the podcasts.
Yeah.
Taking the same episode and
exactly.
just releasing it on all of them.
Yes.
Because I'm not sure. Because what she has to say about, dual science careers and raising kids and so forth, I think is really interesting. Plus, her view of industry is very important from a female perspective. And hopefully she would be, now that she's not at Merck anymore, she could be more honest about it. But
more forthright is a better way to put it.
Yes, whatever. Sure.
Right. I guess I go next. Ryan writes, "Hi, TWiV hosts. Thanks for reading my follow-up to TWiV 468 and for letting me pour out my heart. Or pour my heart out. That's a better way to put it. I take the advice to not beat myself up too hard and just continue working. Prof. Condit asked, at which stage in my career am I? I am currently a postdoc in Saudi Arabia at King Abdullah University of Science and Technology, KAUST. I'm loving the work here, but my contract is coming to an end and my children are starting school back in our native Singapore in January, so I'm currently on the job hunt. I'm a little sad that Prof. Condit forgot about me, especially since I wrote into TWiV previously to express my appreciation of him. Guilt trip. Please see below. Point number 4, highlighted, asterisk, sad face. Best regards, Ryan. P.S. I'm joking, by the way. I can't expect you all to remember every detail of every writer, but I'm glad Professor Racaniello remembered about my love for influenza B viruses. We both did our PhDs in flu B, and that puts us in a special club. Friends wanted to use brotherhood, but that's too bigoted. Really? Okay.
And every B in that email is capitalized.
Every single B is capitalized. That's right, for influenza B.
He's into influenza B, yes.
That's right. Do you want me to read his other email?
No, I don't.
I don't think that's
Just it's there to remind us that
it is a good reminder. And Ryan, I'm sorry I forgot about that. My apologies. But the refresher makes it quite clear. I remember it. I remember it well. In particular, all the capital B's.
Yes.
All right, Rich, you're next.
Aya writes, "Hi there, Twivers. Hey there, Twivers. So the last few episodes have heavily featured critical conversations regarding academia and the scientific publishing industries. And as a master's by research student, I have found it both incredibly insightful and daunting. I do not exactly fit the early stage career perspective Rich was calling out for. However, I do feel my experiences provide insight of what it is like to even get yourself to the PhD stage. I've been thinking long and hard about what I would like to contribute to this discussion and if my experiences would be relatable to other TWiV listeners. So here goes. Before I divulge my story, I would firstly like to thank all of you for fanning the flames of my passion, or obsession as you frequently call it, for virology. I have recently changed my part-time work schedule to the very early mornings, so now I have time to listen to TWiV again. It feels like quite the cosmic intervention and has motivated and reminded me of why I'm working so hard at a time I needed it the most. I come from a large working-class family and grew up in the outskirts of London, UK, and went to average state schools, achieving decent grades and accolades along the way. As a kid, I was always obsessively interested in how and why things worked the way they did. Luckily, I had and still have very supportive parents who accommodated my curiosities as best they could. Think encyclopedias, mini microscopes, endless trips to museums, saying, "Oh, that's interesting, dear," to repeated cool facts. It was during high school that I decided I wanted to get a PhD, admittedly completely ignorant of what happens post-PhD. I wanted and still want to know everything about how, insert problem here, works. Luckily, I have a stubborn determination that has not yet failed me. I studied biomedical sciences as an undergrad, and during those years discovered that viruses were seriously cool and recognized that they lead the way in understanding molecular biology and immunology. With the guidance of a fantastic BSc mentor, Professor Steve Goodborn. Currently, I'm working on dengue virus host interactions, and it is everything I wanted it to be and has me so excited about the research I can do and be part of in the future. Yet, there are still days when I think, what the hell am I doing? I could be earning more money by now if I'd chosen a different career path. But I'm up before dawn working a menial job to get myself through my postgrad studies. That brings me to something I feel has been missing from the conversation. Personal circumstances and how they are treated by the system, shall we say. Whilst we're immersing ourselves in our studies/research, life, with a capital L, happens and can seriously throw you off kilter. Mental and or physical health can take a battering when you're trying to juggle keeping up your grades, bank balance, and social life, keeping your grades, bank balance, and social life high. I'm sure many people can relate to that, whatever stage they're at in their career. The issue for me and for many others is the general attitude to those that cannot keep their personal issues from bubbling over into their work life. Just in listening to people speak about those that are off work for the nth time, you can hear the disdain and judgment, which subconsciously makes me worry that if it all gets too much for me, I will also be judged for taking time out to recover. How I think this relates to all that has been said regarding tenure track, high-impact publications, etc., is stress. In TWiV 468, Amy rightly expressed her frustrations about getting a faculty position, and it really struck a chord with me. These are the things I worry about when thinking, is this really what I want to do? Can I really cope with the constant pressure to be better than everybody else? I will probably never be as good as scientist as Amy. Hey, look, comparison to others. But I still aspire to a career in academic research. My perspective at the moment is, can I face the financial stress to get through another degree and maintain my sanity? Many academic institutions are now publicizing extra mental health support and offering various courses on coping strategies, but all that is worthless if the general attitudes of academics themselves remains the same, judgmental and unsupportive. Anyway, I guess what I'm trying to say is that there are a whole host of non-academic issues many graduate students have to overcome before they can even get to a postdoc level, and the job uncertainty thereafter is a massive source of stress and mental fatigue. Whilst I realize the work week debate is in regards to paid academic staff, I feel it's important to note that many grad students are also relied upon to carry out research for publication, especially in small labs. Many take this on whilst working another job or jobs to fund their living expenses, and that can also impact their productivity even if they are in the lab 40 hours a week. Maybe this is more of an issue in the UK, how well funded are master's programs in the US, but I feel it is important to raise anyway. FYI, I am in the lab roughly 40 hours a week and work part-time 10 to 12 hours per week. Lastly, I think Vincent spoke about the fact that there are no established research technician career pathways, which is a shame, as it would be a great way to keep people in academia that perhaps do not want or have the immense responsibility of running a lab and a small team, but are highly skilled at running experiments, etc. Alas, I'm working on a PhD application as it is still something I want to do with all my heart, but I might have to grow up and get a real job first so that I can afford to do it. With all the negative things I've mentioned, I want to emphasize that I'm still hopeful and optimistic that I can achieve my academic goals. Best wishes, and keep up the amazing work. I'm so thankful for it. Aya, she's a master's student at Bristol.
Right.
Just a couple of things I want to point out. Generally, in my experience, master's degrees don't get a lot of support in the U.S. It's hit and miss. I've seen master's degrees supported maybe with small stipend, but not like PhDs. And in fact, in many academic institutions, the master's degree is looked upon as a way to use graduate programs to create revenue. However, I would hope that in the UK, a PhD is supported sufficiently so that you would not have to have a second job. That's certainly the way it is in the U.S. It's not, it's not a great living, but it's a stipend. It's usually enough to, keep you fed and housed.
That may not remain the case for much longer.
I was just about to say something.
That's a separate issue.
I think the points about personal circumstances is good because we haven't really touched on that. And, as he or she says, it's a stressful endeavor, and I have seen people leave and never come back, and they don't get a lot of support, and that's not fair. Right? So thanks for that.
It's, I see in this a genuine passion and also some concern about where it's going. I don't know, from my own experience, all I can think of is that you have to follow your passion.
Yeah,
totally.
And that is what, in the long run, will carry you through. And if the circumstances and everything else ultimately wind up trashing your passion, then it's time to move. And you don't, you're never going to lose. You haven't lost anything. You won't lose anything. And there'll be a job for you. So try and focus on, they say live in the moment. That's the hardest thing to do. But try.
I want to interject something here. It sounds Pollyannish, but when I was a kid, maybe freshman in high school or something like that. We were told the story of Robert Burns. Robert the Bruce. He was a Scottish leader and exiled in wars and everything, and his army was defeated, and he sought refuge in a cave. And in this cave, he had time to reflect upon his life, and in doing so, he looked around the cave and saw this spider. And the spider was attempting to make a web. And it tried again and again to make the web, and it kept jumping to the next rock, and it kept missing. And it just kept missing. It did this he counted 40 or 45 times. The spider just completely failed. On the 46th try, or whatever that try was, it succeeded. And it built the web. And it caught some flies. And you reach a point where you're at the 43rd attempt to really fulfill your passions and to make your life worthwhile because you really want to do this. And you don't do it because you're going to get the respect of your peers or stuff like this. You do it because it gives you pleasure inside. And that's the passion, obviously. And it's easy to get discouraged because, even if you've got a grant and even if you've got a position, we get depressed every now and then because we reach a dead end in our research, or we reach some stupid academic rule that the university has just suddenly made up that cost us an extra $10,000 that we had in our pockets, and now we don't have it to put forth for our research. I mean, there's a lot of roadblocks and a lot of stones that are put in the way of progress, and yet it's amazing to see the people, and you're listening to them on this podcast. Obviously, everybody here has experienced all of those frustrations in their lives, and yet here we are, 5 disparate people, not desperate but disparate, all not sharing a common background, but all engaged in passionate behavior, and we wouldn't give this up for the world. And that's why this comes across as real, because it's real. And I think what you want to do is real. And so as long as that is the case, and there are so many things that aren't, that's the thing that you should cling to and say, to me, this seems so real that I can't give it up no matter what happens. And eventually you find ways of doing it. And I'm quite amazed to see how many different ways there are of reaching the same goal. How many part-time jobs have I had? How many, turn downs? How many rejections? All these things, they really build up over a while and really get you down. But if you think back to that little spider trying to complete the web, and of course, if he doesn't or she doesn't, whatever that spider's sex was, they don't eat. And the same is true for us. If we don't succeed eventually in our passion, then we don't eat. And that's important too. And to know what point it is to say, I've tried 47 times and I can't complete the web, and now I'm going to have to go out and try to do something else.
All right, Pollyanna, that's enough.
Is that, have I cried enough into this microphone?
I should just add for the record that there's more than one occasion where I felt like, maybe I ought to quit. Yeah.
Oh,
so
almost everybody I know in science has felt that way. So
that's right.
Yeah.
That said, not everybody makes it. I mean,
no, you're right.
Be aware of survivorship bias, and you are listening to 5 people who have succeeded in various ways in science or related endeavors on this podcast. So, failure is an option, but stick with it to the extent that you can, because clearly you're into it.
Yeah, we're not going to hear from people in science who didn't make it and are doing.
We might.
Else,
I don't know.
Not sure they're listening. We'll see. Next one's from Jake. This is the first of the book contest emails. We have 31.
Woof.
Hi, Viral Profs. Please put my name in the hat for the current book contest. I also wanted to add a couple of my reactions to the arc about academic work ethic, being obsessed with science and so on, specifically from the last 2 episodes, 468 and 469. The emotion and frustration and near despair that was in Amy's voice in 468 was so evident and familiar to me. As a postdoc 3.5 years on from my 6-year PhD, I feel absolutely stymied by the academic pyramid scheme, and if I think too much about it, I'll also become incredibly emotional. When I was starting grad school, I was fully on board to be obsessed and to spend 80 hours a week in a lab. Now I'm so jaded and disappointed with the game that I have a very hard time staying motivated to even work a 40-hour week. Honestly, I would be so much more motivated at this point in my life to work an 80-hour-week writing grants, managing a lab, mentoring junior scientists, giving talks, and networking with other PIs than being where I am, stuck doing approximately the same types of work as I was doing 10 years ago as an undergrad researcher, i.e. robotically moving small amounts of liquid around.
Dear.
What I'm getting at is that it's not just ridiculous that science and nature papers are the key to achieving a tenure-track faculty position. It's even more ridiculous that we trainees are expected to suddenly become good grant writers and mentors when the training is 80 hours of wet lab work. All this for 1/3 the salary of our colleagues in industry. I've been listening to TWiX for 5.5 years, and I think those and other podcasts are the only reason I haven't completely lost my mind. Thank you for the new immunology one, by the way. But over that time, I don't feel like any progress has been made in addressing these issues in academia. If anything, they've seemingly gotten worse even before the current presidential administration took office. I don't know exactly how, but maybe someday this podcast can help be a vehicle for change. Maybe if enough listeners and other scientists get on board, we could get organized and figure out a way to produce a better system for academic research. I'm at the point of frustration with the current system that I would definitely be willing to devote the rest of my career to creating a better one. Best, Jake. Well, we would love to be the vehicle. We're here. We talk about it all the time, if anyone.
We do.
But, a lot of scientists are not listening. They're doing what they can to survive, and that's it. They don't want to change the cell science nature situation because they can't. Alan, you're next.
Sure. Joseph writes, "Greetings, TWiV hosts. I'm ready to contribute my non-data-driven and perhaps purely anecdotal thoughts on productivity and hours worked based on my own experiences. I don't agree that working more leads to increased productivity. During my PhD, I worked the typical 80-hour work weeks mostly, which were jam-packed with experiments, time for data analysis, paper reading, more experiment planning, and free lunches at seminars on Fridays. Oh, I love those. The general impression I got from my PI was that those long hours were not necessary to succeed if you planned really well and were excellent with things like time management and focus. But by and large, the more time spent in the lab at the bench, the more data would be produced and potentially lead to more publications. Side note, I think I'm on the John Udell science family tree somewhere, since my PhD advisor, Ike Eisenlohr, was John's first postdoc, or one of his first. So I'm not surprised that this message was or is similar, though I'm sure each has their own spin. I finished with one first author publication in JVI, which I was really proud of because of the amount of work that went into building that project from the ground up. Maybe if I picked a different project, I would have had more first author publications for my PhD studies, but I'm happy with that work. Things began to change when I took a postdoc at a small biotech company called Immunotope. There I worked more modest 40 to 50-hour work weeks. The nature of the work was more translational, more streamlined, which helped with the hours part of the job. And there I was more successful in my publications. 2 first author research papers, a first author review, and a book chapter out for review now. These publications were in journals that favored more translational research and weren't as high impact as some others, but I'm still happy about them. As much as a Nature or Science paper would be incredible, sometimes the fact that the data hold an interesting story is enough, at least for me. In the fall of 2014, I retired my pipettes and culture flasks to become an instructor in biology at Villanova University. My role now is strictly teaching, plus other faculty things like advising committees, etc., and I've even morphed from a viral immunologist to an organismal biologist teaching human anatomy and physiology and histology. Now my work week is roughly 40-plus hours on campus, plus an hour or so at night and on weekends, grading, lecture, prep, etc. If I try to push myself for too long during the week, I can feel my thoughts become cloudy, and I'm more likely to not give my full attention to whatever I'm doing at the time. I've learned to walk away from the computer and lecture preparations to do things that are more meaningful for my peace of mind that are not science-related, though admittedly I do think about work and ways to communicate information to students in more meaningful ways, more than the hours here probably represent. What I've learned over the years is that work-life balance is necessary. There needs to be some mental decompression to stay sharp and focused while you're in the lab or planning, analyzing, etc. I also feel more productive and creative at work since figuring out this work-life balance issue. But boy, I don't know. If I were looking for an academic position as a PI, I'd be working a lot more just because of the competitive nature of science. Ultimately, as scientists, we're pulled from multiple directions and have to find our own middle ground. For me, that middle ground is a less intense work week, hour-wise. For someone else, it could be a more intense week. I'm looking forward to hearing some other views on this. And finally, the weather in Villanova, Pennsylvania today was a nice 59 Fahrenheit, 15 C, and much like Rich, I did not get outside.
That is a wonderful letter.
Yes.
That's a really nice summary of an experience. It's very useful.
Yeah, you have to find your own middle ground. That's what it's about.
Exactly.
Right.
Exactly.
What works for Rich Condit's not going to work for Joe or Dickson or other people.
People used to ask me why I'm taking off to go fishing. I never answered them. I just took off and went fishing. Never regretted a day of it.
You could have had a Nobel Prize if you'd never fished.
Could have. No, I don't think
just kidding.
so. I'm
just kidding. This episode is brought to you by Blue Apron, the #1 fresh ingredient and recipe delivery service in the country. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting the highest standards. Oh, don't yawn.
Stop it.
You can't yawn during an interstitial.
They're not supposed to know. This is not television. I can yawn.
Just kidding. Setting the highest standards for ingredients and building a community of home chefs. Dickson, you like to cook, but you've never used a Blue Apron, right?
No, I'm a scratch cooker.
Wasn't it in Indiana, Kathy? I asked someone, it was Adam Zotnik, what he likes to do. And he said cooking. And I said, did you ever try Blue Apron? I like Blue Apron because I don't have to shop. That's my main thing. It's delicious. It's fast. It's a fun thing to do. And I think if I had kids at home, I would probably say, hey, let's do a Blue Apron tonight. Yeah. That would be fun. Yeah. But I don't anymore. But I think it's a good way to get people interested in cooking as well. And what they do is deliver fresh, pre-portioned ingredients and step-by-step recipes right to your door, which can be cooked in 45 minutes or less. They offer 12 new recipes a week, and you can pick 2, 3, or 4 based on what best fits your schedule. And they send only non-GMO ingredients and meat with no added hormones. Here's their menu. It changes every week based on what's in season. Upcoming items include seared steaks and garlic butter with oven fries and romaine salad, butternut squash pasta with kale and brown butter walnuts. What is butternut squash pasta? Is it made of butternut squash?
If you take a butternut squash and just nuke it, you just take a butternut squash and nuke it for a little while. It comes out like spaghetti.
Got it, spaghetti squash.
No, that's
really
a spaghetti
great.
squash. A butternut squash, you can scoop out the inside.
Yeah, and you mix it with pasta, or is it just on its own?
I don't know exactly what they're doing there. I have a pasta recipe that I do where you scoop out the cooked butternut squash and mix it with the pasta. That's quite good. So it might be that.
I have the feeling that it's integral in the pasta in the same way that spinach pasta is.
It's cooked in. Yeah, that's it.
When we cook butternut squash, we cut them in half, and then we fill the cavity after we take out the seeds and stuff with a combination of butter and brown sugar, and put them into the oven until it softens up, and then we scoop it out and eat it. It's fantastic, just like that. But
all right, barramundi and mixed mushrooms with jasmine rice and Napa cabbage. Napa, wow. Roasted chicken and full vegetables with cranberry and ginger compote. Cooking together builds strong family bonds.
I agree with that.
So have fun together. Take the recipes you love and add your own family twist to them. Blue Apron is treating This Week in Virology listeners to their first dinner a $30 value with $30 off your first delivery and free shipping. Go to blueapron.com/TWiV. So check out this week's menu. Get $30 off your first delivery with free shipping at blueapron.com/TWiV. Do it today. Blue Apron, a better way to cook. So on Pod Save America, they do Blue Apron ads. They do it in the beginning, and that they say a better way to, and then one of the other persons on the show starts interjecting some political stuff in there, and then they say cook. It's very funny.
Right.
Well, we could say a better way to
Sequence,
replicate
Plaque
RNA
assay.
genomes,
Right.
plaque assay, cook. Unfortunately, that's our last Blue Apron ad. So we can't do it in the
So
future.
what is the favorite dessert for left of center politicians?
Left of center?
Yeah, left of center.
Probably that stuff, creme brulee.
No. No. Cling impeaches.
Oh, you're full of jokes today.
Today, that was a bad joke. But wait till next week, you'll see.
And by the way, the non-GMO ingredients is not a particular TWiV position. That's in the ad copy.
That's the ad copy,
That
for
is
sure.
exactly right.
Absolutely. We have spoken out about that before,
We have.
Yes.
haven't we?
Yes, we have.
Right, now we have a paper
Indeed.
which was suggested not too long ago by a friend of the show, Islam Hussein, who you heard from earlier. And
I feel like you might have had some insight into this paper already.
it's funny because he wrote, wow, I didn't know that phages can penetrate the epithelial layers of the body. And guess who the editor is? This is a TWiV paper. And so I looked at it, and indeed I had handled it for MBio. It's called Bacteriophage Transcytosis Provides a Mechanism to Cross Epithelial Cell Layers. The first authors are Nguyen, Baker, Padman, Patwa, Dunstan, Weston, Schlosser, Bailey, Lithgow, Lazarou, Luke, Forrest Rower, who's been on TWiV, Blumberg, and Jeremy Barr is the senior author. You still haven't gotten over that cough.
I've got something.
Maybe you have too many phages in you.
It's possible.
Now,
Did everybody get flu shots, by the way?
I did, yeah.
I got
Oh,
it.
yeah.
I did.
Dickson and I went together.
We did.
Oh, right. Yeah.
We did.
Yeah.
Matter of fact, I got flu block. I was really interested
Ooh.
in that.
I don't
yeah.
think I have the flu. No, I don't have that.
Flu block is the insect cell produced
That's the one that's it's the one that's a recombinant baculovirus.
So this is very cool because the flu block is the only one that has the extra glycosylation site in the HA of H3N2, which is important because when you grow it in eggs, you lose that glycosylation site and it no longer matches the circulating strains. Anyway, what was I going to say? Oh, so this is very cool.
Yeah, bacteriophages.
That's
So
right.
MBio papers. So what I'm an editor, so I get submissions and then I send them out for review. But this one is one of these MBio direct things where I get all the reviews already and all the back and forth between the authors and the reviews, and all the authors say, I'm happy with this, and I get it, and I get to look at it and make the final decision. I think that's very cool, and that's, of course, what
So
they
wait
do
a minute.
at M-Sphere.
If you're unhappy with it, do you get to just knock it even though everybody else likes it?
Yeah, but I'd have to have a good reason to
This
knock
is
it.
true.
If-
This is
yes, Rich, what were you going to say?
How does what's the process leading up to that? Because you got to identify reviewers and stuff, right?
Yeah, the process is, and it's similar for M-Sphere Direct, you identify, you have this paper, there's a form at the journals which tells you how to do all this, but you identify your own reviewers, you send it to them, they tell you what kind of cover letter to write, they send their reviews to you, so it's all, it's not anonymous, obviously.
Okay.
Right? And then you respond to the reviewers, you send it back, and if they're okay, they say yes, if not, you do some more things, you go back and forth, and then the whole correspondence gets sent to the journal, then they pick an editor, and they say, here it is, decide what to do, and you look through it, and if everybody's happy, I'm happy, and so it was accepted here. So
My understanding is that if you as editor had wanted, you could have also solicited
I could.
additional reviews.
Yeah, sure. I think.
And just in the interest of full disclosure, that not only is the editor listed, but those two reviewers are listed
Yeah.
in this gray box as well.
That's right. This article is a direct contribution from a fellow of the American Academy of Microbiology. Solicited external reviewers, lists them here. So I think that's pretty cool.
It basically cuts out that whole middle layer of research editor, triage, send out the reviewers, and
Precisely.
anonymous peer review.
Exactly. That's the good thing, is that you get it reviewed. You don't have an editor saying, do we want to even review this or not?
Right.
I think it's really great. And so there it is. And now for the science. So this
Did you already say it's open access?
It's open. No, I didn't. I did not say where it's from. So these investigators are from Harvard Medical School, Monash University
It's a good place.
in Australia, and
Melbourne.
San Diego State University.
Mm-hmm.
You know, this is very funny. I highlight my PDFs, you know, and then that way I highlight the things I want to talk about on TWiV. And I did this on a different computer, and all the highlights are shifted. They're all in the wrong place.
Oh, so we're going to talk about all the wrong stuff?
A frame shift mutation.
No, I think I could handle it. It's just funny. It's a frame shift.
Huh.
So we have, as you know, lots of bacteria in us, and at the same time, we have lots of phages in us that infect them. And they say here that we secrete more than several billion phages per gram of feces every day. We have lots of phages in us. And of course, they have been found, they can't infect eukaryotic cells. By infect, Dickson, I see you're looking at me strangely.
I'm not.
They cannot replicate.
I know that.
In eukaryotic cells.
That.
Okay. I just could tell you were going to get testy with me.
No.
They can't replicate because, the machinery is different from bacteria and between bacteria and eukaryotic cells, so they can't replicate.
Very different.
But we have lots of them in us because we have lots of bacteria in us, and they've been found in blood and serum. If you give them to mice, you can if you feed mice, you can find phages in the bloodstream, and they end up in all major organs of the body, including the brain. So they can cross the permeability barrier.
Are these antigenic by any chance? I mean, because they're all over.
They are antigenic.
So we should have lots of antibodies.
That's actually something we'll come to later on. It's an interesting point. Mm-hmm. And of course, in the gut, that's where we have most bacteria in our body, and most of the phages are in our guts. Right. And so the question is, how do they get from our gut into the bloodstream, for example? And they say there have been a number of different ways, and one of them is called the leaky gut, where you have damage to your gut, and that allows phages in. The Trojan horse, a phage-infected bacterium gets taken up. Phage display. So maybe ligands on capsids mediate uptake or free uptake by endocytosis. And they say, there's evidence for and against all of these mechanisms. But we thought we would study it in more detail, and that's what this paper is.
There's one more route that you didn't mention that I would be interested in knowing about, the lacteal. The lipid entry point to the gut tract is an open pore, basically. So I wonder if they could gain entrance that way without having to cross any membranes at all.
Maybe. Now they start.
And the question, there is a related question, which is why. It's a why question, but why are they there? Or are they there for a reason? Or do they, by there I mean in the blood, in tissues, et cetera? Because, these are bacterial viruses.
Right.
Theoretically, they've got nothing to do in the blood or in other organs or in the brain or et cetera. So do they have some sort of function there or not?
So the experiment is quite clear. You take germ-free mice and you repopulate them with bacteria lacking phages, and you grow them in an environment with no phages, with no opportunity for the bacteria to be infected. That's what has to be done, okay? So those of you listening, do it. Do that. I'm not going to do it, but I think that's how you do it. You can't do it in people. Now, they have a nice system for looking at this. They make cell monolayers of cells that make tight sheets, like one of the examples is a dog kidney cell line, MDCK cells, very commonly used. They grow these on inserts. They're called transwell inserts. So it's basically a synthetic insert which is sitting in medium. So the cells are growing on this insert, and there's medium above them, which is separate from the medium below them. There's an apical chamber.
You're floating a little cup of cells on the medium.
Yeah,
exactly.
So
it's almost like it's almost like a basket. It's porous in the bottom.
Right.
And the cells grow along. The cells form a monolayer on the bottom of it. There's ways of testing this. If they grow together well enough as a monolayer, you can show that they're not just sort of randomly porous by putting an appropriate dye on one side and showing it doesn't leak through.
Yeah, and they also have electrical resistance, which helps also to show that they're not leaky. Right. It's a nice tight junction. So they use that, and they put phages on the apical or the basal lateral side of the cells, and then they take samples of the medium at different times, and they do a plaque assay.
Right. So apical and basolateral, these are polarized cells.
Right.
Yes, they normally have. They have a top and a bottom. They have a particular orientation. They align with their tops all on the same side. They just do this themselves.
Exactly.
And very tight junctions between them.
Yep.
And so if these were in the gut, which is ultimately what we kind of like to test with this, the apical side would be the inside of the gut where the feces and the phage and the bacteria are, and the basolateral surface would be facing the inside of the body.
Yeah. So they'd have to traverse through the microvilli of a columnar cell, if that's how they go.
Mm-hmm. Kathy, what were you going to say?
I was just going to say that putting these bacteriophages on the cells is really just putting them in the media. And you could imagine gravity allowing the ones in the top to float down. But in fact, in the bottom ones, the bottom chamber, the bacteriophages are just floating free and moving around by Brownian motion, at least until they would settle out. But presumably there are ways that you can flip the chamber upside down and put the phages on, and that's what I was thinking they were doing at first till I saw the picture. It made it very clear.
So they should do this in the space station where there's no gravity, and then what would you get?
Right.
Aha.
I wonder if these things would even settle out over time, or if they're small enough to be sort of kept swimming around most of the time.
Yeah.
That's
I don't know.
a better
Maybe.
question than I thought in the beginning, because if we're going to go to another planet.
Well, tell Kate Rubins. Maybe she'll.
They're added at a very high concentration. We're talking over 10 to the 7th or over 10 million per milliliter. So there's plenty of opportunity for these things to collide with the cells.
And these are phage T4s.
Yeah, I love it.
I thought Rich would like that. What, you're pointing to something?
Yeah,
I have a picture on my wall. Yeah,
you do.
Yeah, so phage T4s have a DNA genome, and they're the ones that some people picture as the typical virus structure, which means they have an icosahedron, but they have a tail with tail fibers.
Tailed phages.
That's right.
This is the little lunar lander phage that.
Yeah. So that's exactly how I think of it.
So they put the phages either in the medium above the cells or below the cells. They wait 2 hours, and then they ask, where have the phages gone? Where have all the phages gone? Oh my gosh, so silly.
And the way they quantitate it,
plaque assay, right?
Is by plaque assay.
It's lovely.
Yes.
Yeah.
They don't come right out and say it, though. They just say by plating with their bacterial host.
Yeah.
Right.
And they move through the membranes, right? If you put them in the top, they move to the bottom through the cells. And if you put them in the bottom, they move to the top.
It's remarkable.
And this is something that happens with different kinds of cells. They've started with MDCK. They test a whole range of cells from different tissues like gut, lung, liver, brain, and they all exhibit what they call bidirectional transcytosis. But there is a preference for the apical to basal direction.
Right.
Which would be going, if it were the gut, from the gut into the body.
Right.
Okay. Or if it were in the lung, I don't know what we're doing with phage in our lungs. I suppose we got bacteria.
Of course, it's there.
Oh, it's got to be there.
Sure.
Yeah, sure.
It would be going from the airway into the bloodstream.
Right.
Right.
So these experiments, the initial ones were done with a 2-hour time point. They then go back and do a time course and look at samples over a 2-hour period. They can detect them. If you put the phages in the apical chamber, they can detect them in the bottom as early as 10 minutes later. And they have calculated a rate of transport, which I don't think we should tell everyone because no one is going to make heads or tail of it, right? But anyway, the rate is dependent on how much you add.
And by the way, this the directional bias from apical to basal is significant. We're talking about at least 1,000-fold.
Yes.
Yeah. So if you're suffering from a bacterial abscess, let's say in your liver or brain, and you had the right phage and the right bacterial species, could you actually cure it this way?
Well, that's a different question, isn't it?
Well, this is
It's kind of a
prelude
related
to that,
question.
though.
It's phage therapy, right?
So if we've got this phage in our organs, and we've got it continuously coming in from the gut, maybe a reason not to interfere with that, a reason why evolution didn't stop this from happening, maybe even assisted it, is that this provides some degree of protection.
Maybe.
Innate immunity.
But Dickson, people are trying to do phage therapy because obviously it doesn't always work, right? People get bacterial infections and they don't spontaneously cure. So they're trying to do that.
If you purposely had 10 to the 31
for protecting
phages,
you.
Yeah, exactly. Right.
Well, there's the experiment to take them away from mice and see what happens,
That's
right?
That's
for sure.
exactly right.
But yeah, in the end, they talk about why this is so. Is this functional or not? And maybe it's somewhat protective, right? They also want to know if any endotoxin present in the phage preparations, which come from the bacteria, could that be causing some damage, which would lead to this? And they purify, they take away the endotoxin, no difference. Right. And the other interesting experiment is they try different phages, right? They try phages across this order, Cordovirales, so order, which is a collection of families, and both phages that infect gram-positive and gram-negative, phages from soil, phages from intestinal reservoirs, phages of different sizes, and they all travel across the cell barrier.
So this adds what I would call rigor to the whole experimental approach.
Yeah,
both this and all the different cell types that they use just with the T4 phage. And the authors did not have to point it out. The reviewers were able to recognize the rigor.
Yes.
This is a comment because I just finished teaching the grant writing class, and people are now starting to put an entire paragraph into the approach section of their grants, pointing out the rigor and reproducibility of their approach. And my understanding is that the reviewers should be able to glean it from the design.
They should. And another conclusion you could draw from all of this is that there's probably no specific receptor for any of these phages in order to go from one side to the other.
How do you think they're being taken up, Dickson?
That's a great question.
Well, we'll get back to that, okay? That's called a teaser.
Should I yawn again?
I would have liked to see all of these are cultured cells.
Correct.
Right?
Mm-hmm. They are.
Can one not make some sort of primary cell?
No, you do it in organoid. You could do better than that even.
Well, you got to be able to make this membrane in the chamber. You got to be able to have a monolayer that's not passively permeable.
No, I hear what you're saying.
Mm-hmm. I'm sure you can do that. Yep. And then probably they're doing it, right? The next thing they did is they labeled the phages with fluorescent dye and put them on these monolayers.
So, and the first label they use is cyber gold, and that labels the DNA, and that becomes important a little bit later, but they didn't say it right up front, and so I was kind of on edge till I figured it out.
You were on edge.
Yeah.
The
had a
apical or the
big
basal?
question mark there.
And from this, they can, once the phages are fluorescently labeled, they can do a number of things. They can look at the proportion of fluorescent positive cells, and they use two different ways to do it. They used epifluorescence microscopy or flow cytometry. Hey, by the way, over on TWiV, we had an email from a listener. He wrote, he said he was listening to them, and Michael Schmidt said, we quantified the number of cells by fax, and he thought, what did they send to fax? How does that work? He had no idea. So he wrote it, and I said, it's fluorescence activated cell sorting. And he said, I listened to that 6 times, and I still couldn't figure it out. Isn't that great?
It's a good point when you throw around terms like that, and we know what they mean, but it's not obvious if you're new to the field.
So flow cytometry is a better term, right? Because it's not an acronym.
Right. We've described this before, and it's fascinating. It's basically a machine that can, one by one, look at individual cells and analyze them for what sort of fluorescence they're throwing off and quantify that. Sure. And even if you want to take sort the cells that have the same color in the same tube and different colors in different tubes. Yes. It's an amazing piece of equipment.
How many colors can you do now at once?
I don't know. Lots.
Yeah, lots.
Like over 10 probably.
Whole visible spectrum.
No, not the whole visible spectrum. It depends on the dyes, the fluorescent dyes you have, right? Yeah. But there are a lot of those. Anyway.
This must have been, I remember being exposed to this in the late 70s. I think that may be when it started. So it's been around for a long time.
Sure.
Important technology.
Have you ever seen the way they quality control for potato chips? I'm not joking. I see it on how it's made.
How do they do it?
Well, there's a jet of air that comes out when the camera detects a defect. And it blows the chip off of the line of potato chips. And
if it's too small? You mean if it's too small?
Yeah, or they're too big or broken or something like this. And the cell sorter works the same way. As soon as a fluorescent cell comes out, boom, a little jet comes out and blows it into the tube.
I think we're messed up. I'm sorry.
No, there's a lot of similarities here. People get their ideas from all kinds of places.
It's a capitalist, free market driven economy. Whatever people want, they get, right?
You know what? And sometimes they get exactly what they don't want.
So by these two methods, they find the proportion is either 10%.
That sure digressed in a hurry. Sorry.
10% or 1.7% with the two methods. Okay. So there's anywhere from 1 to 10% of the cells are taking up phages depending on what method that you use.
Right.
All right. And they talk about that, but I don't want to talk about it. I don't think it's very interesting. They say the phages, the signals are either discrete puncta or diffuse. I love this. Diffuse fluorescent clouds. So, most people talk about the cloud and it's either the sky or where your data goes up.
This is true.
But now these are phage clouds, fluorescent phage clouds. Isn't that great? And they say some of them are within membrane-bound vesicles.
Up there in cybergold space.
And others are free-floating within the cytoplasm. I just picture a tailed phage floating so gracefully in the cytoplasm. All right, then they want to know where they are, where these phages are. So they took a cybergold positive cell, and then they processed it for ultrastructure inspection using correlative light electron microscopy.
This I just love.
Can you explain it?
This is great.
Neat.
Well, I can't explain how it's done. Maybe one of you can.
No,
but,
I can't.
But it's you have the light microscopic issue. In this particular case, is a fluorescence micrograph, but it's optical. It's done with light waves, and you see a normal micrograph, which has the sort of magnification where you can see one or two cells in a field. Mm-hmm. But you can't really see in a light micrograph all of the ultrastructure that you would see with an electron micrograph. And this technique somehow allows you to do transmission electron microscopy on exactly the same image.
Yeah.
So that you can line them up precisely and you can correlate for example, where your fluorescent signals were very precisely with the ultrastructure that you see in the electron micrograph. So you might, for example, in this case, see a spot of green on the fluorescence image and look in exactly the same place on the electron micrograph, and lo and behold, there is a membraned vesicle. And you can say with confidence that green correlates with that membraned vesicle.
Now, I'm no expert on terrific. This, but yeah, my understanding is that this is one instrument.
Yes,
so
I believe that's correct.
It's built so that you put your slide on the stage, and you've got a confocal microscope and a transmission electron microscope looking at the same stage, and you, I guess, take a photo of the confocal and then start doing your transmission electron microscopy, which then scans across this same thing without moving anything.
So when you read the figure legend in 4, they talk about the data in panel A are maximum projection between the 37th and 43rd optical sections, 3 microns to 4.2 microns above the cover slip. And then in B, they're from the 47th resin section, and then they tell where that is. So I had the impression that it was at least 2 different sections, maybe, that they're looking at. And it may be in the same machine, like you said, but maybe not the exact same plane. I could be wrong.
Yeah, I'm not sure how the alignment works, because the confocal, you're going to have to align. I mean, the idea is that you want to look at the same plane, but it may be working on different alignment scales. Mm-hmm. Right. So I'm not sure. It would be interesting to hear from the authors or from somebody else who's done this technique, how they align that in the depth dimension.
I'm finding one image here on the internet of how this is done that has two different machines.
Right. It can be done with two different machines, but in reading up about it before this episode, apparently Leica and others are now combining these into one unit. Okay. And I don't know which the authors used, but of course the advantage of having one unit is you don't then have to find exactly the same cell and all that.
Right.
Unfortunately, when they did this initially, they didn't co-localize the phages with the cyber label. Right.
That was the result. They didn't find that.
They didn't find that.
Co-localized. Right.
Yeah. So then they went on and repeated this with dual labeled phages, with the DNA labeled with cyber and the capsid linked to a CY3. Is that cyan? CY3. What color is CY3? That doesn't.
Looks like red in the image.
Red. And there you can see those two dyes early. They do a time course early on. These two dyes co-localize within vesicles, and then as you increase the incubation, they dissociate. So the DNA and the protein dissociate, which seems to me that's the right thing, right? To happen.
Well, not unless you think that the phages are actually
maybe if they're coming apart,
replicating, or if they're coming apart. But I think they have another explanation that has more to do with the stability of one of the two labels and
right,
and so forth.
Dyes. Anyway,
right, so there's basically two possibilities. One is that the phage is coming apart, and the other is that somehow the label is degrading, that the phage is still okay, but one of the labels is dissociating from the phage and wandering away. And the problem is that if the phage are coming apart, that's inconsistent with having
infectivity on the other side.
Infectivity on the other side. Though this is a matter of numbers.
Numbers, sure.
Okay, you could have 99% of the phage coming apart in the cells and 1% getting through.
Right.
And that may be enough.
Exactly.
Sure.
So the infectivity assay is very sensitive, right?
And you wouldn't see by these sort of visual assays, you wouldn't detect, you wouldn't be able to quantify 1% that stayed intact and got through.
Right. So in the end, we can't co-localize label with phage in these experiments by looking at them, correct?
Correct. Which is something to consider.
Yeah, I mean, they say we need to do more work on this. The next thing they ask is, in these cells, where is the phage? So they fractionate the cells into different compartments, and they look at phages, quantify the phages, and they find that they were found in all of these subcellular fractions.
Including the Golgi.
In particular, the membrane-rich fraction.
The membrane-rich fraction, but not in the nucleus, right?
No.
Right.
Not in the nucleus, but throughout the cytoplasm.
And their images show membrane vesicles inside the cells that have phage-like looking things in them. Mm-hmm. And some of the dye also, although dissociated from phage, winds up in membrane-looking things. So the sense is that this has to do with some sort of membrane activity. And I mean, what I picture is these things being gobbled up by endocytosis and then transported through the cell and spit out the other end by exocytosis, with maybe a few weird membrane pathways in between.
But lysosomes are not involved in this?
Yeah, well, that's a good question. Usually, well, it depends on the particular endocytic pathway. There are multiple ones, right? Clathrin-dependent, caveolin-dependent, independent. And so, not all of them may intersect with lysosomes. But yes, if it did, you would think it would be degraded, wouldn't you? And
exactly.
It wouldn't come out. But as Rich says, it may be maybe a lot of them are degraded, and if you just get 1% out, then you pick up that infectivity. So they use a series of inhibitors to try and track where in this transcytosis pathway the phage is going in. They use Brefeldin A, which inhibits post-Golgi membrane traffic and protein translocation, and that inhibited not completely, a little more than 50%, I think, and 38% of the phages were transcytosed in that treatment compared to a control, but no inhibition when they used wortmannin, which is an inhibitor of receptor-mediated transcytosis, bafilomycin, an inhibitor of endosomal acidification, no inhibition of transcytosis, chloroquine, which is also an inhibitor of endosomal acidification, or an antagonist of calmodulin, which inhibits membrane transport, endocytic membrane transport, essentially. So
my understanding is that Brofellin A is kind of a membrane wrecking ball.
Yeah, it is. It is. Okay. Also inhibits polio replication. So they say this suggests that they may transit through the Golgi apparatus and then they're exocytosed, but they need to do more work on that.
Right.
The last part is an estimate of how many phages enter our body on a daily basis.
Right. So based on this is purely an in vitro study, but if the gut is doing things at the same rate, then...
I think, Dickson, we could do an experiment. We could feed you 10 to the 10th T4 particles. It's not going to hurt you.
No,
then at different times afterwards, we could take your blood.
You could do that.
And then see how, the kinetics of phage in there.
You could do that over my dead body.
But that's done.
I'll bet you Dickson's already got a pretty high background of T4 in his blood.
In his blood,
sure
probably. Yeah.
I'm sure he does.
Are they infectious, Rich?
Sure.
Yeah, probably. So they use numbers, a number game here. So concentration of bacteria in the gut, 9 times 10 to the 10th phages, bacteria per gram of feces. It's a lot. Then they use literature references estimating how many phages would be there. Okay, 5 times 10 to the 9th phage particles per gram. So 2 times 10 to the 12th phage particles within the colon of an average human, which excludes you, Dickson, right? Because you're not average. Right?
That's right. That's because I'm a member of this show. No one's average here.
Then they use their experimentally derived rate of transcytosis against cells, gut cells. They estimate that the average human body transcytoses 3 times 10 to the 10th phages per day.
Billions and billions.
That's right. That's right.
It's a lot of phages.
It's a lot of phages.
And as we're sitting here, Dickson, they're just transcytosing.
They are.
That.
I can feel them.
They're flowing into you.
I can feel it.
And they say that's transcytosis, and they say the leaky gut model is probably not correct, because if you think of these numbers, to achieve this number of phages, this would require 256 square millimeters of damage in your intestine, and which they say was probably not be good. You would not feel good. We would have a lot of inflammation.
More than a centimeter squared, right?
No, but remember the way the gut repairs itself, or actually the way it grows. Each gut cell, each columnar cell, lives about 7 days. Starts in the crypt, works its way up to the top of the villus, and then it dies and is extruded from the top of the villus. And so what I would like to know is there's a moment in time when that membrane is open and then it closes as the cell egresses and closes it up. Is that the moment that these phages can actually get in then?
So, there are M cells in your gut that are professional transcytosing cells, right?
M cells.
They're always sampling the gut.
You bet.
And they could be, but according to them, a lot of different cell types are involved. By the way,
do M cells and other gut cells transcytose stuff that is as big as a phage?
Like cells
that's a good question.
I love the cells of Pan-F.
I mean, it is thought that reoviruses are taken up that way, but these are bigger.
Yeah.
But, I guess it's possible because you feed mice phages, it ends up in their blood.
There you go.
So I've been trying to think of some kind of a control where you did the same experiment with some sort of particle that was sort of phage size and has no biological significance whatsoever, some sort of nanoparticle or something.
Yeah, you could do that. Sure.
To see if these cells will just sort of randomly do this with almost anything.
Yeah, I think that's a good idea. It's a good experiment. You could make particles of different sizes and see what's the upper limit.
Yeah,
they have them.
You could swallow a baseball and see if it gets transcytosed. Kathy, 256 square millimeters is 2.56 square centimeters, which is about an inch.
Wait,
right?
if it's about 12 millimeters by 12 millimeters, I figured that would give you 244 millimeters squared.
Yeah, that's 2.56 square centimeters.
12 by 12 would be 144, wouldn't it?
You're right. I did 100 error. Sorry.
But anyway, the point is that
was a gross comment. That was a gross comment that you just made, Alan.
Right.
as Dickson
just trying to keep things in order.
As Dickson points out, if you spread that out over the whole surface of the gut, in transient mini micro pores, you could probably tolerate that and get a lot of phage through.
So really now, let's speculate. What, if any, is there a purpose of having all these phages in us, assuming they're in us? Which
are they killing bacteria on the other side?
Yeah, well, they say that maybe they prevent intrusion of opportunistic gut microbes. That could be. So I think we did a paper.
The germ-free animal is the answer here.
Rich, do you remember the Penn State TWiV and Allen, too? Rower was talking about phages getting caught up in mucus and defending against bacterial infections.
Right.
And he's on this paper, of course.
Yeah. And that was really cool. Yeah. Phages stick to mucus, and mucus lines mucous membranes, obviously. So bacteria that try to get through the mucous membrane and access to the cells below have to run the gauntlet of phages.
That's a good one. Running the gauntlet of phages.
Pamplona.
Very good.
The phages of Pamplona.
Nice. Well, I think the germ-free mice can help with these experiments.
That's true. But I remember Dr. Morris Pollard always telling us that
Notre Dame.
You can assure that there are no bacteria there because you can't culture anything. And then, of course, that's not true anymore. And then he says, but you can't rule out the fact that they have viruses because they probably do.
So they also talk about this possibly being a way of horizontal gene transfer between phages and eukaryotes.
There you go.
But it could also be between bacteria and eukaryotes if you consider the phages being transducing phages, right?
Yeah, sure. Definitely.
Yep.
Well, I'm going to be the cynic here because with absolutely no disrespect to the authors or the study, I think that it's quite likely that none of this has any purpose. Okay. That the phage just, there's a lot of phage around. Okay.
Possible. Yeah.
And if just a couple of fractions of a percent get through, they're going to show up in your blood. It's just kind of litter. All right. And may have no purpose at all.
So where are the antibodies that'll trap those and get rid of them?
Well, they say that they may be at low enough levels that it's not enough to raise an alarm, or they lack co-stimulatory factors such as endotoxins.
Or they're so
tolerant.
There's tolerant.
Since these have been around through our entire lives for all of our generations, we may simply build tolerance to them very early on. These are self.
Yeah.
Mm-hmm. Yeah. Because you're born with phages, right?
Yep.
We did that study from a former student of Kathy's, right?
Mm-hmm.
Born to be phage.
Born to be phage. So many titles.
The baby's microbiome, yes.
All right, so I tweeted to Seth Bordenstein today, is there any good evidence for phage DNA in eukaryotic cells? But, he has eukaryotic DNA in a Wolbachia phage. It's a paper we did here on TWiV. And Dickson, probably you weren't here for it, I would guess.
I don't remember that one.
Judging by your puzzled look. But, and of course, theoretically, if there's so many phages in us, horizontally it could be transferred. But I'd like to know if there's any really good evidence for it. And he hasn't answered, though. Yeah, all right. Anything else? Yes.
I have two comments. Since this is open access and people could look at figure 2B if they wanted to, this is where they put on different amounts of phage and note that the amount that they collect coming through is concentration dependent or dose dependent. And neither you nor the authors talked about what does this mean. Mm-hmm. And I tried to figure it out, and now I'm trying to think about it in light of the idea of maybe some of them getting degraded, but I couldn't quite picture why, what is the barrier that to the excess ones in the high concentration or why don't, I guess, if you put on a lower concentration, why don't they all get through?
Often you do that kind of experiment. I'm thinking mostly about enzymology here, but often you do that kind of experiment to find out if there is a specific process that is saturable.
Mm-hmm.
Right.
Okay. And in this particular case, that doesn't appear to be the case, at least at the concentrations that they've tried. It could be that if you go to a higher concentration or something like that, you can saturate it. But that, in my mind, that's consistent with the notion that we're not talking about a specific receptor or something like that.
Right. Okay. And the other thing that I just thought was interesting, I made a note of it at the end of the section where they talk about that they need to do further work. This is still in their results. Further work using real-time microscopy, repeating the Clem analyses with dual fluorescent labeled phages is required to further elucidate how phage transcytosis occurs. And I wrote, they didn't have to prove the mechanism to get this published.
Yeah.
So.
Good. That's good. All right. Let's move onwards. Thank you, Islam. And also Saul Silverstein mentioned this paper to me. He said to me, have you seen this? And I said, I think so.
Good thing.
I think so, yeah.
Well, I'm glad.
Right, so we had 31 entries for the book contest. We won't go through all of them, although we should get to some of them in the future because some of them have interesting information in them.
That's right.
We've read the first one already. That was Jake.
We have read the first one.
Follow-up.
And here's the second one is from Steen. Your contest and end date happens to be my official start date at Rutgers. I hope to make a pilgrimage to the TWiV studio sometime.
Mm-hmm.
Now, Steen has written often about plant viruses. He's been working as a postdoc. He's got a job at Rutgers now.
Good for him.
Good for you.
Actually, Steen, how many hours a week do you work?
Yes.
How many cell papers do you have? How many nature papers do you have?
So Rutgers has a great ag school, so I think maybe that's the connection here.
Well, I'd like to, this sounds like a success story to me.
Yeah.
I'm interested in the details.
That's right.
The next one is from Islam, who said he entered the contest, said, TWiV gives meaning to my life. It's lovely. And I'll take one more, and then we can switch to some others. It says, Basel writes, well, actually, it's a listener pick, so I'll just wait for that, right? Yeah.
Yes.
I'll do one more. Tom writes, does it help that I'm a Patreon supporter? Didn't think so. Been listening since Vincent was on Futures in Biotech, May 2009. TWiV and TWiM are almost always of interest, but TWiVo is my current favorite. The podcasts stimulate my imagination like science fiction, but the more disciplined thinking helps my analytic capability. Now, with 100% confidence, I say pithy things like, you can't get flu from a flu shot, vaccines are only for serious diseases, and you should wash that with bleach. I have a podcast called Function. It's about art and engineering, but we also talk about office supplies and stationery. FunctionPodcast.com.
By the way, that
office supplies and stationery.
His first sentence triggered a response that I think we can build on.
Wow.
It says, does it help that I'm a Patreon supporter? And
you know what that means, Dickson?
I do. Of course I do. So stop patronizing me like that.
No, I'm picking on you.
You're picking and patronizing. So what if we did offer books for, like if you could donate X dollars, right, we have a book for you.
Otherwise, stop. It's already there. Those are called rewards. And if you ever looked at the page, you would know it.
Well, then why doesn't he just increase his Patreon support? He gets a free book.
There are different levels of rewards, but I won't go through them now. They're like $1 a month, $5.
Is the book one of those rewards?
No, it's not.
Well, let's make it.
I always have one copy of it. But things like, $1 a month gets you your question to the top of the queue. $5 that gets you a mug. $10 gets you a T-shirt. I think $100 gets all of us to go to your hometown and do a TWiV, something like that. Or gets you to listen in on a TWiV. I don't remember.
That's right. We'll come in and cook a Blue Apron dinner for you.
But if you want, we can do a parasites book for one of those levels.
Well, we can get as many books as you can handle.
I understand. Anyway, so Alan, take a couple now.
Okay, Trudy writes, not often that I've actually read the TWiV paper before you guys discuss it on the show. Neither is Dickson. Oh, who am I kidding? It only happened this once. Anyway, it was a pleasure to listen to TWiV 468 with Amy Rosenfeld. I'm very fascinated by the Zika story and love how quickly our understanding of this virus has progressed over the last couple of years. And she's a follow-up to Vincent's pick of Nick Lane's book about oxygen. She recommends a book by Stephen Johnson called The Invention of Air. which chronicles the story of Joseph Priestley and his discovery of oxygen. And Stephen Johnson was the author of The Ghost Map. She says, probably one of my very favorite microbiological detective stories. I had the pleasure of seeing Stephen Johnson give a talk at Emory many years ago. I must say he's almost as gifted at telling stories as Dickson Despommier.
Well, that's very kind of her to say that. And by the way, Amy went to Emory for her undergraduate, so that's a nice connection.
Now, let me ask you something, Dickson.
Please
Does
do.
Trudy ring a bell?
She should.
She sat next to you.
Because Quasimodo rings a
She
bell.
sat next to you right there.
The name Quasimodo rings a bell.
Yes. Trudy sat next to you
Thank you.
in the taping studio.
Indeed, she did.
Sven writes, quick email to try to win the book and a couple of small comments regarding episode 468 about papers and Nature Science Cell. I'm on the old continent and never lived in the States, so not sure what the situation is there. I can guarantee you here, everybody does not count Cell and Nature papers when receiving an application, even though it may be because I'm working in a more environmental field where the pressure's lower. When Amy said something like, you guys had the opportunity to change things but did not, I need papers in big journals to get jobs. I find that a bit hypocritical. It's easy to blame others for not changing the system. Everybody can make a choice to go for open access. A good CV can have several other strong assets besides just the journals cited in it. Okay.
Depends on the institution.
Not here in the U.S.
That's sorry, Sven. Here on the new continent, that is not definitely
That's right.
not the case.
Lost in translation.
And then I forgot who said having these top journal filters helps to select through hundreds of applications. Well, yes, nobody ever said it would be easy, but if one wants to bring down the awful system on which scientific careers are based right now, everybody will have to do their bit. Selection can also be shared among several people, or the description of the requisites can be more specific to lower the amount of applicants. There are definitely other ways to make selections. And I would certainly agree with that. Unfortunately, those all require change, which has not been forthcoming. He adds, cheers from Bergen, Norway, where it is freaking cold and dark.
Exactly.
But beautiful.
Yes.
All right, Kathy, take a couple.
Number 8, Kevin writes, hello, Twivsters. I don't really have anything to say. Just wanted to enter the contest. All
That's
best
fine.
wishes, Kevin. Number 9, Marianne writes, hi, Twiv team. As always, happy to listen along during my commute. Also very happy to see the launch of Immune. If you ever need a researcher in the front lines of cancer immunotherapy to sit in, let me know. I did have one quick follow-up regarding the last episode where I think Alan mentioned that biotech for PhDs is basically a PI position in a different setting. I'd really like to respectfully disagree. I'm a VP, Vice President for Research at a biotech company in Canada. I think I'm better suited to my research position in biotech than I could ever be as an academic, partially due to my wide area of interest and my varied research background that never quite allowed me to find my niche. During my training, I moved across very different fields, often because I always followed research I could be passionate about, and I was never intimidated by entering a new area. My current position requires me to be up to date in many different areas of research, including immunology, virology, microbiology, parasitism, a big reason why I keep current in all the podcasts, as well as being versed in business, quality, clinical research, and investor relations. My background in science outreach has served me very well, communicating our science to the public. I often refer to what I do as a jack of all trades, master of some, and this is exactly what I find exciting about the potential of research. I would feel stifled by following a single line of research that was dictated by funding opportunities, which I think some people may find ironic, as there is a perception that all companies by nature have very narrow research aims. Generally, people don't quite understand the quality of basic research in small and medium biotech, as they assume all discoveries are made in academic labs and all drugs are developed only by big pharma. It kind of misses a big space in between where a lot of cool things are happening. Cheers, Marianne.
I'd like to apologize if it came across as meaning that these are exactly analogous positions. I think what I was trying to say is that biotech for PhDs is similar to a PI position in the sense that you're moving to a research environment and being placed in charge of a certain amount of research. I'm quite familiar with the differences between these two environments. I talk to a lot of people who are on the biotech side, And there's a different breadth to it. There's a different feel to it. There's a different and the situation at biotechs is different from the situation at big pharmas versus medium-sized pharmas. So yes, there's a tremendous diversity of experiences within those. I think what I was getting at was that these are similar in the sense of scope of authority and relative prestige.
All right. 10 is from Anthony. 11 is from Johannes in Australia. 12, Dickson.
Andy writes, Dear Dr. R and team, first of all, thanks for all the work you do on the Twixt series. I've been listening for around 5 years now. I started when I was getting my Master's of Science in Immunology of Infectious Diseases and found your podcasts an engaging and entertaining way to see some things from a more microbe-centric view. I'm now nearing the end of my PhD at the Royal Veterinary College in London, UK, looking at how the microbiota affects the outcome of streptococcal infection in a zebrafish model. As I am more of an immunologist by previous training, the discussions and papers on your podcasts have been great for me. Anyway, as you can tell by the subject line in this email, I was motivated to write in because of the virus's textbook giveaway. I reckon we'll one day come to appreciate the virome in a similar way we do the microbiome. This book would really be great to brush up on and expand my virus knowledge. Thanks again for everything. Best wishes. Okay, Robbie writes, hi, that's number 13. Robbie writes, hi all, I'm writing in for the book giveaway. The December 1st deadline coincides perfectly with my PhD applications being due and gives me an excuse for to procrastinate a little bit more. Thanks,
That's
Robert.
right, December 1st.
That's true. And I'll do the next one too. 14. E. Brian writes, write whatever you want, in quotes. Thanks for the opportunity to win TWiVarium. Keep up the good work.
Yeah, that's what we said, right? Whatever you want.
We did.
Mm-hmm. Rich.
15, Richard writes, TWiV Docs. Thank you for the great episode on Zika research. I enjoyed Dr. Rosenfeld's assertive views on journal publications. It demonstrates the diversity of views and the reality of surviving in academia. From my perspective, research and the academic competition for advancement can seem more like a knife fight than a collegial refuge for independent thought. Thanks to Dr. Amy for your candor and the incredibly complex research on Zika and its complications. Hope I'm the randomized winner of the book. Rich is from Culver City, California.
And he has a DDS.
Cool. Take the next one.
Yeah, he's a dentist, right. 16, Sarah. Hope I'm the selected number. She's Sarah's a Master of Public Health and a PhD and a postdoc.
In Gainesville.
She's in Gainesville. Holy cow.
Do you know this lab? Lednicki lab?
Oh, yeah, of course.
Uh-huh.
Take number 17.
Okay. 17, John writes, hey, TWiV teams. Now, that is HEJ, which is Swedish for hi. I know that. Hey.
Hey.
I'm writing this email whilst listening to you talk about getting a career in research is so tough. I'm a third-year microbiology undergraduate student from Sweden doing my degree in Glasgow, Scotland. Of course, I feel worried about getting myself a career within research, but what concerns me even more is how badly we, the students, are informed about how competitive it is. I do realize that it might scare many of us, and so less students would aim at a research career. However, I believe that there can be a backlash where students with huge loans graduate and expect jobs to be available. Instead, they're hit with immense competition, resulting in even fewer taking the fight and going into research. I have to thank you, as I did not realize how tough it is until I started listening to your podcast, and I am still aiming to go into research simply because I find it so incredibly satisfying to be part of new discoveries and expand on our knowledge of the world. University of Glasgow has given me the opportunity to do a work placement in industry for a year, and I have just been accepted for a placement at the Pasteur Institute in Paris. I'm very excited about that. I wonder if you might have any advice for me to make the most of my year working alongside researchers. Thank you for the great podcasts, which makes procrastination not feel as bad. As always, a pleasure working for you.
Listening to you.
Heart. That A with an O over it. It's a peculiar Swedish vowel. Hort, I think that is. Yeah. Keep up good work in Swedish.
So,
you got a lot of great opportunities here. My advice would be to forget about all that other stuff and just enjoy what you're doing, because Pasteur Institute, a little while in industry, you're in a good time of your life where there's not too much pressing down on you. My biggest mistake in all of this was worrying too much. In retrospect, I could have done all the same stuff without nearly the stress, just by, getting control of my head. So enjoy it. That's my advice.
And while you're at the Pasteur Institute, make sure you visit the crypt of Louis Pasteur. It's fabulous.
Is that like the crypt of Lieberkuhn?
No. That was a ridiculous joke at this point.
I think it's quite sciencey.
No. Have you ever been?
Yours.
Did you ever go to Louis Pasteur's crypt?
No.
It's fabulous.
I've been to the crypt of Lieberkuhn.
Well, then you've got a gut level feeling about all this. Then
that's a good joke. That's a good joke. All right.
Ba-dum-bum.
Let's skip the rest of these mails. Let me just tell you the short ones where they just said, I want the book. Number 21, Alex is, I want to win the book. Number 22 is from London. Number 23, Seamus, Eve from London. 23, Seamus is from Wash U in St. Louis, a postdoc. 24, Temet is from Ohio State University.
The Ohio State University.
The Ohio State?
Yes, that's part of the name.
State. That's right.
Okay. Columbia University in the city of New York, that's the name,
yes.
But I just say Columbia University. 26, Robin is from Helena, Montana.
That's a good place.
Department of Public Health, cool. 27, Kevin is from Duke Microbiology. 28, Michelle is from the Australian Army Malaria Institute.
Interesting.
How cool.
Interesting.
29 is a big letter. 30 is a big letter. 31 is a big letter. Okay, so
back on Michelle. She's currently in Georgetown University in the O'Neill Institute for National and Global Health Law.
Right.
Cool.
Fulbright scholar, right? Yeah, so that means she'll go back to Australia. So we had 31. We'll read some of those longer ones next times, but we had 31.
Yeah.
And let me do a random number generator between 1 and 31. Is everyone ready?
Yep.
We are, except
ready.
I just wanted to mention Scott's intro. It's wonderful. Dear TWiV Ninjas. I like that. We've never been called ninjas before, but I like it.
I'm pushing the button. The winner is... winner's number 8. The person who had nothing to say.
Ta-da!
Number 8, Kevin. All right, send us your address, twiv@microbe.tv. And for you others who did not win, we are doing the same giveaway on TWiVo.
Oh.
So this week, we're going to record that TWiVo. It's not recorded yet, so you can't enter yet.
Wow.
I guess you could, but you don't know what to say, right?
Exactly.
So just check out TWiVo, and that's a way to get more people listening to TWiVo, right?
That's fine.
But we'll give it the same book, and it's inscribed for a TWiVo listener by Michael Cordingley.
Yeah.
So check that out.
Yeah.
And to all of you who entered, thank you. We will read your longer letters because they have wonderful thoughts in them.
True.
So,
and comments and things.
And the thing I really like is, first, people are all over the world listening to this, which is so cool, doing all sorts of different things.
Right.
They like it. So that means we're doing the right thing to a certain extent, I suppose. And
globalizing
it's very fulfilling to hear all of our listeners, who we don't often hear from, right? True. All right, let's do some picks of the week. Alan Dove, what do you have for us?
I have a little app that I got maybe a month or two ago. And I've only used it a few times because we have a lot of light pollution around here, but it is just a really cool use of the phone technology. This will work on, there's an Apple version and an Android version. It's called Planets. And you fire this thing up, it uses your GPS position on the phone and the orientation that you're holding the phone in from the accelerometer in the phone. You hold it up to the sky at night, and it gives you a sky map. So it will show you where the stars are. It'll outline the constellations for you if you like. And you can just pan it around the sky, and it's like this little magic box that gives you labels on all of the
Is this
stars
GPS based?
Yeah.
Okay.
Yeah, so it uses your GPS position, and then the position you hold the phone in determines how it orients the screen. And it seems to work. As far as I can tell, I haven't put it through its paces exhaustively, but
and it labels stuff.
It labels stuff.
This is great. This is
right.
Just
so
great.
you can look up and you can say, "Oh, well, that's that's Aries and that's Jupiter over there," and
because normally I recognize two constellations.
I can find a
Big
couple of
Dipper
things
and
in the sky,
Sagittarius,
and
right?
Big Dipper and Orion.
That's Sagittarius.
Right.
Yeah.
That's
so
it.
this one evidently is free. I've had one called Star Walk for a long time, or Star Walk 2 that costs a little bit of money.
Going to get it right now.
Yeah. Yep.
This is cool. Oh, this is great.
This may have had ads on it, and you pay a couple of bucks to remove them, or something like that. But
that's cool. Yeah,
I actually have a folder on my phone that's got called Universe.
Universe,
and it's got Star Walk and Moon and Night Sky, Sunrise and Sunset, the Space Station watch, NASA TV, and another NASA site. How to add this. Star Walk works okay.
Wonder what other things could use labeling in a phone like that besides stars. People. It'd be cool if you could hold your phone up and know who was there. I mean, if they had a phone, it should be possible, right? Of course, privacy and all that stuff.
Yeah, face recognition.
Gosh, that would be so cool. Dickson, you understand what I'm saying?
Yes.
Okay. Rich, what do you have?
Oh, my goodness. What do I have? I have a series of 3 little animations that were sent to me by my buddy Nassim that were produced by NPR called A Short History of Humans and Germs. And it goes back to the beginnings of time and talks about coevolution of people and microbes and the good news and the bad news. Winds up with vaccines and takes you on into the future. It's entertaining and well done. A couple of little glitches I noticed, but I'm not going to gripe about it.
All right, Dickson.
Well, I have a YouTube 5.5-minute video of a fantastic person. His name is Levon Biss.
This is so cool, yeah.
And
it's very cool.
He wanted to get an insect photographed in complete focal planes so that you could almost look around it. So it looked 3-dimensional. And the way he accomplished this, the video shows how he does it. He uses a macro lens for a microscope, and he focuses on about 1/30th of the insect at a time and does a through focal plane. He takes about 10,000 images.
He does slices of it.
He does slices. Right. That's right. Optical slices. And then he pastes them all together. And then he pastes all of the 30 together to create the insect. But that's the most important part of this, is that the printer that he uses to make his exhibition was the size of the room that we're sitting in right here. And it's an incredible process that they have 2 technicians that do the dust off as the printer head passes one side to another side of the matrix that he's printed on. And you end up with an insect that you could almost walk into. It's so big when they finish the printing.
This print is like 8 feet long of this fly.
It's in Oxford University's Museum of Natural History, and they have the second largest insect collection in England. And you think, well, who's got the first one? And of course, it's the British Museum of Natural History. And who do you think contributed most of those insects? And it was Charles Darwin.
Is that right?
And he's got pictures of some of the original insects collected by Charles Darwin.
Cool.
And I bought the book, by the way. The book is it's not better because it's smaller. They have no fold-outs. No, like Playboy, they have no fold-out sections for these insects, which they should have done. But that would have been too expensive, I think.
What are you going to do with this book?
I'm going to show it to my grandson. I'm just going to regale him in detail.
Kathy, what do you have?
Well, as Dickson wrote to me last night, I have something that's similar but in the opposite direction of dimension. So it's a feature from National Geographic where they've captured the president, which is the 3,200-year-old sequoia in Sequoia National Park. And so they talk about all the statistics about it, and they show some of the pictures that went into making up the final total large image of it. But what's really cool, too, is they have a video that you can watch. And then scrolling through and looking at it on your computer, you can see the whole tree. And it's just beautiful.
It is.
It's enormous.
So this picture they have here is the picture, right?
Yes.
And there's a guy standing in the top.
Those guy.
Yeah.
And there's a guy at the bottom.
Tree ornament.
Too.
They look like tree ornaments. That's how small they look.
Why
They're
did they
too
get
small to be tree ornaments. They're like ants.
Why did they get to be in the tree? That guy.
Well, that's part of how they took
by
the
scale.
pictures and go.
To give you a scale
yeah,
feeling.
give you scale too. If you go about 4 or 5 pictures up above that, you see 3 guys hanging from the tree.
Yes.
Or there might be women. You can't really tell at this distance.
Right.
President is still growing at breakneck speed. Wow.
That's right. Some of the great landscape painters of the 17th and 18th century inserted people into those pictures to give you a sense of scale. And so that's the reason. That's the history of that.
3,200 years old, right?
Yep. They'll probably turn it into mulch for gardens when it dies.
I vote for that tree as our president.
Hey,
Yes.
Good idea.
certainly more intelligent.
My pick is a New York Times article called "Laptops Are Great, but Not During a Lecture or a Meeting." This is by Susan Dynarski. And
With whom I vehemently disagree on this point.
well, the point is that she feels that if you take notes in a classroom on a laptop, you're not going to learn as well as if you write them out by hand. And she cites a few studies that support this. Those selectively interprets a few studies to rationalize her bias. No, so she doesn't allow students in her classes to use electronics.
There was an extensive discussion on Twitter about this. There are a lot of problems with this position.
Well, I think the studies are probably not definitive, right?
No, first of all, one of the main studies she cites has been very, very widely cited, and I think maybe 1% of the people who cite it actually read it. But the consistent theme in this is in these studies, they're all experimental settings where people were assigned to either the laptop or the no laptop group, and they didn't get any instruction on how to take notes on a laptop. And so there was a tendency by a lot of people to just transcribe, which of course is a terrible note-taking technique. And lo and behold.
But that is what students do, Alan, because the problem don't learn how to take
laptop.
Notes.
The problem is not the laptop.
Well, it is because they can type faster than they can handwrite. And if they were forced to only do handwriting, then they would learn to take notes. And then, yes, maybe they could graduate back to a laptop. But if they can type as fast as they can hear people talking, they're going to type everything and not learn from it.
But I've
seen it in class.
I get that. No, I get that totally. I've seen it in class too. But the feedback mechanism for that is called a grade. Right? And if you're flunking in your classes and it's because you're not keeping up with what's going on in the lecture, you need to work on your note-taking skills.
So
that's not how they do it, though.
So at medical school here, they give you a handout, which is a collection of the PowerPoint slides with spaces to write down below each one. And as the lecture is given, you're supposed to write into the bottom of each slide.
I've been to presentations that are like that. I mean, the reason I feel so strongly about this is one of the central skills of my job. And over the past 10 years, when I started doing this, it was really just not feasible to lug the heavy laptops of the time around to interviews and to conferences. And so I didn't. I took notes by hand in a system that I developed in college. And I made the transition about 10 years ago to using keyboards, which I've found vastly more efficient for my work because, again, I evolved a system that works for taking notes. I'm not sitting there transcribing. The studies on this are, I think, very skewed in that they are randomizing people to groups based not on their experience with this or their background in note-taking, but ordering them to take notes in a method that they may not be familiar with, and then saying, gee, it doesn't work well. A couple of the other studies, one actually the students who were using the laptops were required to bring up unrelated information on the screen, and they found that kids sitting next to them then retained less of the lecture because they were distracted by the screen, which, yeah, no kidding. So there are ways to deal with this that do not involve a blanket ban on laptops. I think that's an extreme measure and not an appropriate tactic.
I think you should choose whether you want to use a laptop or not. And if you're not going to use it right, it's your fault. You should be responsible for what you're doing. But to ban it, I think, is absurd. The reason I picked this is because I don't ban laptops or any electronics in my classroom, but I do tell everyone all the lectures are recorded.
Right.
And you could just sit and listen and then go later and take notes. So a lot of people don't sit there and take notes on their laptops. But what I do find interesting is that a lot of them have tablets, and they have the slides there, the PDF, and they write on the tablets, with the panners. And and they say, well, this is writing, so this is better. And that's kind of neat too. If you don't have the laptop note taking down right, you can still write it. So that would be banned in her classroom. You wouldn't be able to use a tablet either, which is not really fair. So it's an interesting issue. And
so I teach an undergraduate course now, which I'd never done before, and I demand that they bring their laptops because we look things up as we talk. And I want them to go to the internet, and I want them to look up things, and I want them to communicate with each other, and then I have them mail that website to everybody else. That's something I'd like you to know about, and I use that laptop as a teaching device, not as a note-taking device. Because you can enrich every lecture this way if you use them.
Well, I don't really want them looking up things during a lecture. If it's a discussion course, I could see that, right? But they're not going to listen to you if you tell them to look things up, Dickson.
No, but we have a discussion. We
that's fine.
Do this more as a discussion.
That's fine.
Yeah.
By the way, I've seen Alan at conferences. He's got his little. You have a tablet you use with a keyboard.
Yeah, now I actually use I use my iPad, and I have a separate keyboard with it. And I can put that down in my lap so that the clacking doesn't bother anybody.
Sure. And Alan, you're absolutely right. The grades will tell you whether this works or not. And everybody has a different system.
But she's not the only person to ban electronics from classrooms,
of course not.
Right?
But as another issue with this, which people railed about on Twitter, is there are students with various types of physical or neurological problems that prevent them from handwriting. And she even says in the article that those folks are allowed to use laptops, which of course immediately stigmatizes them.
Yeah, right.
Which they didn't need any more of, since they're already, kind of facing a lot of issues, and that is another argument against an outright ban.
Yeah. All right, we have a couple of listener picks. We have Basil's from above. The Zika-focused TWiV episode is timely, as I was recently reviewing a histopathology slide of Zika infection in the mouse, and he gives a link for a whole slide scan, free access, courtesy of the Joint Pathology Center. This is the Armed Forces Institute of Pathology. So you can go check that out. I'd like to take this opportunity to introduce you, as well as many listeners, to the Veterinary Systemic Pathology Online and Wednesday Slide Conference. These are amazing resources, free to access, but hold tremendous amount of data and knowledge within them, not to mention the countless hours spent on making and maintaining the two resources. So these two links, very interesting stuff. Lots of slides.
Cool.
And then John writes, since I'm biology training impaired, I have difficulty with some of the terminology in the Twix family of podcasts. Leaving aside, if just for a moment, the Fantastic Parasitic Diseases 6th edition that I lean heavily on for TWIP, I have found the following biology books from the OpenStax collaboration at Rice University to be very helpful for techniques, technology, jargon, and pretty pictures. They're available via Amazon or freely available in PDF and are peer-reviewed. So he has for biology a book for science majors, Concepts of Biology, a book for non-science majors, Microbiology for non-majors, Anatomy and Physiology for a course for life sciences. They also offer textbooks for many other subjects. OpenStax is philanthropically funded by the Bill and Melinda Gates Foundation and the Hewlett Foundation, among others, and provide tutor and online material in addition to the textbooks. OpenStax.org/FAQ. Thanks and regards, John in Limerick, where the weather over the weekend was wet and windy, 50 millimeters of rain and gusts of 130 kilometers per hour, thanks to Atlantic Storm Brian. Those are very cool resources. OpenStax.
It's really a cool resource. I looked at some of these books. They look great.
Very nice. All right, that is TWiV 470, Apple Podcasts, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. We will have more book contests. I have a lot more to give away, although that one was autographed, so that's pretty cool.
Very cool.
If you like what we do, consider supporting us financially, microbe.tv/contribute. Dickson, Despommier, parasiticdiseases.com. TheLivingRiver.org is pretty cool, too.
It's great.
Thank you, Dickson.
It. You're welcome. This is very rewarding.
Was this a good one?
This is excellent.
Did you say they're not all good?
I didn't say that.
Yes,
Some are better than others, but they're all good.
You said some of them suck.
You know, you're picking on me again, Vincent. And you know what? They're going to say, don't pick on Dickson.
Yeah, I have some of those later.
Don't
coming
do that.
I'm not picking on you. I'm just,
Well, you're trying to rile me up.
It's like
I'm easily riled.
The Odd Couple.
Oh, no, it's not. No.
No?
I'm much neater than you are.
No, you're not neat at all, actually.
I'm very neat.
I am neat.
You should see my fly boxes.
I did.
You did. And they're very neat.
Montana.
Very neat.
Kathy Spindler's at the University of Michigan, which is in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiV, and the sponsor of this episode, Blue Apron. Thanks for your sponsorship for the past year. We'll now have to forge ahead without sponsorships.
Oh,
Talk about that.
To learn how to shop for food for a change.
I know how to shop for food. I just don't like to do it. Hey, you go and shop and get a sandwich at night. Come on.
No, I don't.
Yeah, you told me I'm going to get a sandwich the other night.
That's at night, but I don't do that much. We shop for our food.
I don't like shopping.
We enjoy it.
Introductory music by Ronald Jenkins, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.